Planting protocol. Set up your workspace with designated materials, including 3 pieces of filter paper. Next, check the plate label for the appropriate seed number and record each number on the corresponding filter paper, starting with number 116, 75, and 56. This is done to prevent any future confusion in the remainder of the protocol. Our next step is to determine how many seeds for each line number we need to plant. In this experiment, we plant 3 seeds per line number. So to speed up future planting processes, we sterilize all the seeds at one time. Therefore, we'll be sterilizing 9 seeds for line number 116. The seeds that we are working with have been placed in 1.5 milliliter microcentrifuge tubes and labeled with the corresponding seed number. After the correct seed line number tubes have been matched with their corresponding filter paper, the designated amount of seed is poured out. Because the size of seed we're working with is so small, tapping the tube with the side of your finger or tapping the top on the table may help in the removal of some of the seeds. We will now sterilize the seeds with an ethanol and seed sterilizer solution. First, we will take a sterile pipette tip and place it on our 100 microliter pipette, set to 50 microliters. Then we'll dip in a seed sterilizing solution and saturate the seeds on the filter paper. Then, using a different pipette tip, we will saturate our seeds with 70% ethanol solution. Our next step is to line up the labeled auger plate on the designated planting template. To begin the planting process, we dip our planting tool in a microcentrifuge tube filled with water. This makes it much easier to pick up the small seeds. Gently pick up your specified seed and place it on the surface of the auger. Be very careful with this step, as we don't want to damage the auger in any way. After all 9 seeds are planted, make a vertical black line on the outside of the auger plate using a permanent black marker. This is done for alignment purposes in the scanning protocol. After placing the lid on the auger plate, secure the bottom of the dish by wrapping parafilm around the outer edges of the plate. Finally, place the planted plate in the refrigerator to let the stratification process begin.